Okay, so good morning, students. Let us start today's class. So let me share my PPT again. Okay, so yes, is it visible? Am I audible? Can you see my PPT and can you see me?
Yes, sir.
Yes, sir.
Perfect. So let us start the class. So I'm just taking the continuation of a morning class, okay, which is tissues of anatomy. Anatomy and physiology goes hand in hand, okay. There we are going for studying the cell structures or the morphological features will be studied in the anatomy, where the functional features of the corresponding organs or systems or tissues will be studied in the physiology. So let us just extend the morning lecture here. So yeah, so coming back to the topic, so we are discussing the topic of types of cells tissues in the human body, and now I would like to appreciate the Stever actually. So Stever ignited with many questions, so that is very nice. Okay, he's asking me many questions after the class, and he's asking how to differentiate a prokaryotic and a eukaryotic cell. And he's saying that in the Google it is giving different answers, like sometimes Google giving answers of a single cell or a single cell and multicellular. Prokaryotic single cell, sometimes multicellular, even sometimes even eukaryotic cells Google stating like single cell. So that I will discuss, and let us see again this is cell origin of cells and their story. It is a really interesting story. Okay, so let us see again what are these cells and tissues. Then of course we will go and discuss in detail about tissues. Okay, get ready, that is a very big story. Okay, the cells, types of cells is another big deal. Anyway, let us see these cells again. So can you see this one, my laser, this red color laser on the
Yes,
screen?
sir.
Okay. Okay, so morning we discussed about the cell, then we discussed all these things. Now I want to tell you a story of how a prokaryote is evolved to eukaryote. What is the lineages that prokaryotic cell took in the development of this evolution? Okay, so first I want to tell one important thing, students. This prokaryotic cell is a simple cell. Okay, this simple cell, they are bacteria actually. Prokaryotes are bacteria. So bacteria are divided into two regions. Okay, one is some of the bacteria evolved to withstand extreme conditions. Like some bacteria evolved to withstand in the high temperatures, maybe above boiling temperature, like 101, 99, 100, 101 degrees centigrade. Okay, some bacteria has evolved to live in extreme acidity and alkaline conditions. They can able to live in acidic environment, and they have the capability of living in alkaline environments. Some bacteria can live in extreme cool temperatures. So such extreme organisms that can able to live in extreme conditions, they are called extremophytes, or technically speaking, they are classified as archaea. Okay, these organisms are called archaea organisms. So our prokaryotic cell is evolved into two categories. One is bacteria. Bacteria are normal prokaryotic cells. Okay, they will generally live in a normal environmental conditions. But there are another branch. This branch has took they adopted to the extreme conditions. It can be extreme temperatures, pH, or even presence or absence of water and oxygen. Okay, aerobes and anaerobic conditions they can able to withstand. Such organisms are called archaea. So archaea is one important kingdom, and bacteria is one kingdom, phylum. Okay, phylum. Then another one is eukaryotes. How eukaryotes evolved? The story of eukaryotes is very beautiful. I will tell how eukaryotes evolved from prokaryotes. Meanwhile, let us try to see the major divisions of eukaryotes. So eukaryotes means we understood that eukaryotes have the tendency to become multicellular things, right? Say for example, one is fungi. So fungi is one eukaryote. Okay, what is the role of fungi? Fungi will do the true saprophytes. Okay, they are earth scavengers. They will feed on dead. All dead bodies will be removed from planet Earth because of fungi. Fungi will only feed on decaying
Dead and
organic,
decaying.
yeah, decaying organic debris. Okay, so if there is no fungus, if we are dead, our dead bodies will be everywhere. But we are not seeing our dead bodies, right? Our dead bodies are rotting, they are decaying, right? The reason for our bodies to decay is because of our fungi. Fungi can able to digest even hairs and nails. Okay, fungi is the very good saprophytic organism. Then the second category is animals. I need not to say much about animals. You already know whatever living organisms, the animals we know, like fishes, reptiles, aves, mammals, bilateral cetarians, everything. Sea creatures to fishes to everything are animals. Okay. Then plants. So other third important branch of eukaryotes is plants. So in plants, we have many types of plants like angiosperms, gymnosperms, seedless plants, non-vascular plants, thallophyta. So all these are plants. Okay. Then there is a branch of eukaryotes which has taken to live in a single cell state rather than going for multicellular state. This particular eukaryote has decided to live in a single cell. That means a single cell will do all the functions of the body, like respiration, digestion, excretion, everything, all these three metabolization, everything will be done by a single cell, single eukaryotic cell. Such animals are called as protista. Protista, even though they are a single cell, still they are eukaryotes, they are animals. Okay, in one way or other way. Most of the parasites are our protista. Okay, like your malaria parasite, Taenia solium, Taenia saginata, this Giardia lamblia, Ascaris, Nectar americanus. All these parasites are protista. Okay, so this is all about the eukaryotes, bacteria and archaea. Then I said, right, how eukaryotes have arisen from prokaryotes. What consequences lead to the arise of multicellular organisms on planet Earth? Okay, that I will discuss. Very beautiful story, actually. So whatever we know, all these things, we need to give a thankful to the Charles Darwin. Because of Charles Darwin meticulous understanding of this theory of evolution, today we have understood how the animals are evolving, how new species are coming into existence from pre-existing animals. Charles Darwin never discussed about origin of life. Keep in mind, he never said how life came to planet Earth. Rather than he only discussed about how speciation occurs, how new species will evolve from the pre-existing animals. Okay, his book is called On the Origin of Species. It's not about on the origin of life. Okay, many people has criticized Darwin, that Darwin is going against God. He is telling how life came into Earth. He never said about life. Actually, he said about how the pre-existing animals have speciated into a different species. Okay, on the origin of species. Very beautiful, extraordinary, no, elegant theory it was. Wonderful theory, the theory of evolution by on the origin of species. Okay, by natural selection. Very good. Yeah, now let me go further. Yeah, so as I stated, our common ancestor, that is earliest living organism, has taken three divisions, three types of adoptions. Some early cells have evolved to survive in extreme conditions. They are called archaea. And some has evolved to have a simple life. Okay, they are bacteria. And other one evolved to eukaryota. Okay, eukaryotes. There we have all our animals. Okay, so animals, fungi, plants, protists, everything is in eukaryotes. Now the thing is, so these are the six kingdoms of animals. Okay, we have animals, planta, fungi, protista, eukaryote, these things, basic things. So this is how the animals has been, the cells has been, or living organisms has been classified. So we have prokaryotes, they're single cellular, they will fall under a category called monera, whereas eukaryotes can either live in single cellular state or multicellular state. If an eukaryote is in unicellular state or single cellular state, such animals are called as protista or parasites. Whereas a multicellular eukaryotic cells are taken three important divisions like fungi, plant, and animals. Okay, so still I haven't answered the question how a eukaryote has been evolved to a how a prokaryote has been evolved to eukaryote. That I will discuss. Beautiful story. Same story. Okay, this thing. Yeah. So what happened is in the beginning we have a simple prokaryotic cell in a ocean. So what happened is during the time of during time periodically this eukaryotic cell folded itself, and there are some early adoptions of eukaryotic cell. It started folding itself, and that is what happened. So the prokaryotic cell started folding, and it given rise to some intracellular complexity. It given some endoplasmic reticulum. Because of development of this foldings, these foldings became a part of endoplasmic reticulum. Now within these foldings we can able to store the proteins. So there are some early adoptions has happened within a cell, but the major event happened is something really beautiful. Listen to me very carefully. So there are many types of prokaryotic organisms on the seabed. We have one particular prokaryote which is taking it is taking sunlight by chloroplast, and it is able to generate energy with the help of sunlight. It's taking sunlight, it's taking environmental glucose, and it is metabolizing the glucose by taking sunlight. That prokaryotic organism is a chloroplast. So it is trying to live in the ocean bed by the using energy from the sunlight. Okay, that is chloroplast. Then second organisms are some bacteria like organisms. They are using this glucose. They're going for glycolysis, and the end product of glycolysis is pyruvate. By glycolytic mechanism, they are able to generate 2 ATPs. So bacteria is trying to generate 2 ATPs. With the help of these 2 ATPs, they are trying to survive. And their excreta, I can say excreta, that excreta is a pyruvate. Okay, pyruvate is the excreta of bacteria. Now, we got a third type of prokaryote which has evolved to digest the excreta of the bacteria. What is the bacterial excreta? It is a pyruvate. Now, this is a immobile creature, immobile prokaryote. Now, what this immobile prokaryote did is, whenever a bacteria is excreting pyruvate, the pyruvate is landing on our this prokaryote, and now this prokaryote is started eating the pyruvate. It started metabolizing the pyruvate, and it has generated 32 ATPs. This third type of prokaryote started utilizing generating 32 ATPs, but it is a immobile. It is not using that energy for any mobility. Simply, it understood how to digest a pyruvate, and it got a huge energy, but it cannot do anything. Okay, that is the mitochondria. So mitochondria, chloroplast are the prokaryotes only. They are the earliest living organisms. They are living freely in the ocean. So one day what happened is, there is this mitochondria, an individual organism. So one day a bacteria ate a mitochondria. It eaten a mitochondria. Now after eating, something happened. The bacteria got a superpower. You know what happened? Immediately bacteria didn't digest the mitochondria. Keep in mind, there is a symbiotic relationship. So bacteria ate a mitochondria, and now mitochondria given bacteria 32 ATPs, and that 32 ATPs will be very useful for bacteria to move fastly. Okay, so that is the benefit bacteria got of more energy to mobilize. Now, what is the benefit our mitochondria got? Previously, mitochondria is an immobile stationary organism. So until someone excreting on it, it cannot get food. If no one excreting, it will die. But now mitochondria got a protection inside a moving organism. So they both started living together. They started living together, and such a friendship is called in evolution symbiotic friendship, symbiotic relationship. Now we got a two cells containing single organism. That is the primary primitive eukaryotic cell. A bacteria with a mitochondria became a complicated
primary
cell. That is a eukaryotic cell. That is how eukaryotic cells has evolved. See, so we got a bacteria. Bacteria started eating mitochondria, and this became a preliminary, eukaryotic cell. Such a wonderful concept. So this concept is called as endosymbiosis theory. Okay, so this theory has been proposed by a scientist. Her name is Lynn Margulis. She is the best living scientist when she's the best candidate in the field of evolution to give such a extraordinary groundbreaking theory called endosymbiotic theory. If endosymbiotic theory is true, then an eukaryotic cell containing a mitochondria or chloroplast must have their own DNA. Because we are endosymbiosis saying that mitochondria is once upon a time it's an entire organism. That means it has its own DNA. and somehow another DNA containing organism eaten it. Then mitochondria, even though it is a cell organelle, it must have a DNA. And that is what our link Margulis proved. You know that mitochondria has its own DNA. A cell has mitochondrial
Yes, sir.
DNA, nuclear DNA, even chloroplast. Even chloroplast has its own DNA.
Yes, sir.
How those cell organelles got their DNA? Because once upon a time they are free living organisms, but they left their free living status and they started working together. Okay, so this is called the endosymbiotic theory, and the best proof to show that endosymbiosis is correct by showing the presence of DNA in mitochondria and the chloroplast. Presence of DNA in mito and chloroplast indicates that endosymbiosis theory happened. Okay, see, that is why today we have mitochondrial DNA and chloroplast DNA. This is because these are free living organisms once upon a time, and they have what they started
Engulfed by
yeah,
bacteria or prokaryotic cell.
and they become eukaryotes. Yeah, so yes.
Yes, sir.
So by this, I just want to introduce you one of my favorite scientists who is our Lynn Margulis. Unfortunately, she has expired in 2005, but the contributions that she has done in the field of evolution and especially in the field of endosymbiosis theory is really outstanding. Something she has given a new view to the Charles Darwin on the origin of species. Darwin haven't understood how the complicated life evolved on planet Earth, how single cell came to multiple cell, but uh, uh, she has filled the gap between the missing link between the prokaryotes to eukaryotes by her beautiful theory called endosymbiotic theory. You know, this is called endosymbiotic theory by Lynn Margulis' contribution. She got many awards, students. She got many, almost 180 awards she has in Wikipedia. You know, she is one of the best scientists so far in the history of women in science. Such a groundbreaking theory she has given. Of course, there is some criticism on Lynn Margulis that she said that HIV evolved on some non-evolutionary theory. But yes, but overall, she did some meticulous work in the field of this symbiotic theory. Okay, so by this, I can start our today's class. Okay, so now we have understood how eukaryotic cells has evolved from pre-existing prokaryotes. Okay, because they have engulfed and they have started this symbiotic relationship. So symbiotic is the ground we all
Mutual relationship.
mutual understanding. See, and
Yes,
that
sir.
see, our ancestors followed the motto of understanding each other and teamwork. And we must take a moral, we must take a lesson from that, that even today as a humans, we must work together. Working together can give beautiful outcome like the way we seen in the evolution. So let us stop quarreling each other on stupid reasons. Let us start showing compassion on each other, loving each other, caring each other, working together for the benefit of humanity and for the benefit of entire planet Earth, to the ecosystem and to the nature and to everything. So with this message, I want to start today's class. I think class has become more philosophical, but this is nice. Yeah. So yes, so we are discussing about the cells and tissues, right? So we came to this one, right? So cells become tissues, tissue will become organs, organs will become organ system, and we got a living organism. Then we have very simple 4 types of tissues. We have epithelial tissue, connective tissue, muscular tissue, and nervous tissue, right? So shall we start discussing in detail about epithelial tissue, students? I will take almost half an hour class. Okay, shall we go in depth on epithelial tissue? So yeah, so I guess we can move forward to discussing in detail on epithelial tissue. Okay, so I'm moving forward. Okay, let me do one thing. Let me go slightly depth on individual tissues. Then we can start epithelial tissue. Okay. So usually epithelial tissue is the tissue that is present on the outer membrane of any organelles or tissues or anything, right? Now, this epithelial tissue is broadly classified into 3 types. Okay, these 3 classifications is further divided into 2 types. I will tell, listen, depending upon the shape of the epithelial cell, depending upon the shape, this epithelial tissue is divided into 3 types. They are squamous cells, simple squamous cells, simple cuboidal cells. That means they are in cube shape, or they are in columnar, like a rod, columnar cells. Now, if they are only in a single layer, then they are called simple squamous epithelium. If they are in cuboidal, then it is called simple cuboidal epithelium. If they are in columnar, like cylinders, then it is called simple columnar epithelium. Okay, cuboidal and columnar epithelium. Now, if they are more than a single layer, if there are 2 to 3 layers, then they are called as stratified. More than one layer is called stratified. Okay, so here we have stratified, that means more layers, stratified. What type of cell? Squamous epithelium. So this is stratified squamous epithelium, this is stratified cuboidal epithelium, and this is stratified columnar epithelium. Do I started recording this session, students? Let me check it.
Yes, sir. Already started.
Yeah, fine. So now you got it. Single layer is called simple squamous or simple cuboidal or columnar, whereas multi-layer is called stratified, stratosquamous, stratified cuboidal, or stratified columnar. Now there is a third type of tissue which does not look like a okay, it is only single layer cells, but it looks like a multi-layered cells. Okay, even though it is a single layer, but it resembles like multi-layer. Such tissue is called as pseudo-stratified epithelium. This pseudo-stratified epithelium is only one layer, but because of presence of columnar and mixture of these 3 cells, looks like it's a stratified, but it's just a single layer only. So that is why this is called pseudo-stratified epithelium, looking like a double layered. Okay, pseudo means simply false. Okay, false double layered epithelium. This is a pseudo stratified epithelium. Okay, so these are the types of epithelial tissues we have. Then coming to the connective tissue. So connective tissue is broadly categorized into 3 types: cellular connective tissue, fibrous connective tissue, and ground substance. So coming to the cellular connective tissue, all our white blood cells, and there are other cell, students, which we haven't discussed in hematology, but there are like fibroblasts, macrophages, mast cells. All these cells together can be stated as a all blood cells are a fluid connective tissue. Okay, this blood cells together can be stated as a fluid connective tissue. Then we have different types of fibrous network architecture among the tissues, and that fibrous network is called as fibers. We have reticular network, reticular fibers, we have elastic fibers, and we have collagen fibers. Collagen fibers present in your blood vessels, or all normal skin tissue has the collagen fibers. Imagine collagen fibers like a sari, a cloth. To this cloth, the cells are stitched. Okay, so collagen is a common cloth. On this cloth, the cells are stitched. So that is the collagen fibers. Elastic fibers present to the elastic tissue, like your ear, your lungs have this elastic tissue. Okay, then reticular tissue, reticular fibers are present in between. I forgot that name. Pubic symphysis. Yeah.
Pubic symphysis.
Yeah, pubic symbiosis. That means
Yes, sir.
2 bones will fuse together by a fibers. Those fibers are called reticular fibers. Okay, your ulna and radius of your hand, they are binded each other by some fibers. I forgot the name of those fibers. Okay, those fibers are reticular fibers. Because of presence of these fibers, we can able to rotate our hand. See, my bottom hand is not moving, but up it is moving. The reason is these 2 ulna and radius bones are together stitched by some fibrous substance. They are called reticular fibrous substance. Okay, reticular fibrous substance is further divided. That we can see in detail when we are studying connective tissue. Then third and last and final will be the ground substance. Okay, ground substance is like a fluid state. It's not in cellular state or in a fibrous state, rather than it's in a fluid state. So that can be stated as a ground substance. One of the best ground substance will be mucopolysaccharides. Mucopolysaccharides is one of the best ground substance, which is present in most of the mucous membranes and mucous parts of the body. Okay, so mucopolysaccharides, gluco, I forgot the names. We have some ground substances. Okay, just remember. So these are the types of connective tissues.
Glucuronic acid.
Glucuronic acid, yeah. Glucuronic acid is one ground substance, correct. So these are the types of connective tissue. Then last and final will be muscular tissue. So muscular tissue, again, we have three types. We have cardiac muscular tissue, skeletal muscular tissue, and the what is the third one? I'm unable to see it here.
So smooth muscle.
Smooth muscle cell. Okay. So smooth muscle, skeletal muscle, and the cardiac muscle are the three types of muscular tissue. And last and final, the fourth type of tissue is the nervous tissue. So nervous tissue, what is the primary function of nerve? Nerve is to conduct impulses, nerve impulses, right? Electric impulses throughout the axon. So the cells that can able to conduct electricity, they are called as the true nerve cells. Why I'm telling this? Because there are other cells which are also a part of central nervous or peripheral nervous system, but they won't conduct any electrical impulse. The cells that won't conduct electricity, even though they are presenting in CNS or PNS, those cells are called as glial cells. Glial cells is a type of connective tissue cells. Glial cells, what is the role of glial cells is they will glue, okay, gluing cells. They will connect these neurons together. Say, for example, imagine an electric wire without a plastic coating. So if I have two electric wires without plastic coating, what will happen? I will get short circuit. I will get a very, the electric impulses will they will interfere each other, and I won't get an outcome. My bulb cannot on. Okay. Similarly, that is why we need to insulate the wires. We need to insulate the wire by, usually wire will be insulated by plastic coating. Whereas in the cells, the neuron, the axon will be the axon is insulated by a layer of lipids that is called myelin sheath. Okay. This myelin sheath is synthesized by a cell called Schwann cell. Schwann cell is responsible for providing the myelin sheath to the nerves. So what is the significance of myelin sheath? Myelin sheath will eliminate any possible interneuron short circuits. Interneuron short circuits can be, or that interaction can be eliminated with the help of having proper myelin sheath. Now, there might be some conditions where this myelin sheath will be destroyed. In that conditions, we will have a permanent spasms to the patients. Patients with permanent spasms like this, we will see, right, that permanent spasms are occurring because of demyelination of axons. Okay, axons of the neurons. So yes, there are many types of cells that are supporting the normal function of the actual nerve cell. Like a Schwann cell will give the myelination to the axon, whereas microglial cells will provide nutrition to the neurons. Then satellite cells will ensure the microenvironment of the neurons. Then we have ependymal cells will secrete cerebrospinal fluid. So there are many types of glial cells. Okay, all these glial, oligodendritic, satellite, microglial, Schwann, astrocytes, ependymal cells, all these things are called neuroglial cells. Okay, so yeah, the neuroglial cells is a type of connective tissue. So this is a complete overview of the four major types of connective tissues we have in our human body. Okay, so a quick revision of all types of four types of connective tissue, tissues in the body. We have epithelial tissue, connective tissue, muscular tissue, and nervous tissue. Epithelial tissues are broadly classified into two types depending upon the cellular shape and the complexity of the cell. If the cell is in squamous cell, cuboidal cell, columnar cell, if they are in double layer, then they are called stratified squamous cell, stratified cuboidal, and stratified columnar cells. And there are a type of cell that looks like a
pseudo-stratified.
Yeah, double layer cells, but they are only unicellular layer cells.
Yes,
they are called as
pseudo-stratified.
Then we discussed about connective tissue. So connective tissue, we have three types. What are the three types of connective tissue? We have cellular connective tissue, which is the blood fluid connective tissue. Then we have the fibrous connective tissue. So fibrous connective tissue, we have three types. Can anyone tell me the three types?
Elastin, fibroblast, and reticular.
Very good. Elastic, fibroblastic, and reticular connective tissue, fiber connective tissue. Then third one is, what is the third one? Let me check it. The third one is a ground substance. So ground substance, you have macromolecules and our
Yes, sir.
glucuronic acid are the
Mucopolysaccharide. Yes, sir. So
yeah.
that was glucosamine you were trying to speak. Glucosamines.
Glucosamines. Yeah,
Yes,
glucosamine,
sir.
not glucuronic acid, glucosamines.
So
Yeah.
yes, glucuronic acid and also glucosamine. These are two different. Only a presence of a nitrogen at one position. NH2 group in the glucosamine, and in the glucuronic acid there is a, I think, carboxylic acid group present.
Okay, thank you, Aman, for giving me information.
Thank
Yeah,
you, sir.
yes, yeah. So yeah, they are the types of connective tissue, ground substance. Okay, then we have muscular tissue. So the three types of muscular tissue are cardiac muscles, skeletal muscles, and smooth muscles. Cardiac muscle present in the heart, skeletal muscle present in the skeletal system, and smooth muscles
Colon.
present in the intestinal colon. Okay, now the way that we can able to differentiate a smooth muscle and skeletal muscle is presence and absence of striations. Striations will be absent in smooth muscle, whereas striations, cross striations are present in the skeletal muscle. Whereas a cardiac muscle is a branched muscle. It has branches, and it also has something called intercalated discs. In this particular picture, we are unable to see intercalated disc, but the prominent feature to identify a cardiac muscle will be the identification of the intercalated disc among in between the muscle. Okay, intercalated disc presence indicates that it is a cardiac muscle. And also cardiac muscle will have numerous mitochondria. Presence of many mitochondria is also an indication of cardiac muscle. Okay, then the third one is the fourth one is nervous tissue. Nervous tissue, as I stated, the neuron is a single functional unit of the nervous system. So here it is a basic structure of neuron. It has a head. Okay, head has dendrites. Dendrites will cross communicate with other neurons. And the soma. Soma means the head. Head is called soma. Okay, cell body. It has a
Cytone.
nucleus.
Yes.
And the cytoplasm is called cytone. Then we have the neck. Neck is extended to an axon. In the axon, we have Schwann cells. Schwann cells are responsible for this, myelin sheath synthesis. Then what is this nodes of Ranvier? Why we
Sir,
have?
yes, sir. Sir, the axon where the myelin sheath is absent.
Okay, so I will tell you very simply. I hope you know the difference between a alternating current and a direct current. DC current has a problem. DC current is even though it is a not harm a safe current. The problem with direct current is it has a high resistance. We cannot send a direct current on a 10-kilometer wire.
Yes, sir.
The reason is it will generate more heat, and that heat will reduce the overall efficiency and the voltage of the electricity that is going here. Now, if we start, if we start oscillating the direct current, high peak and low peak, high peak and low peak.
Yes, sir.
The rate of propagation, the rate of enhancing the efficiency of the electric conductivity within a copper line or within any, these channels can be increased exponentially with the help of presence of this, deviations up and down. And that up and down effect within a nerve cell will be facilitated by having these nodes of Ranvier. So the electricity will jump from one node of Ranvier to the next node. So by jumping of this electricity, it giving us a phenomenon just looks like an alternating current. So because of this alternating current mode of mechanism within the neurons can be facilitated by our node of Ranvier. That is the significance of having this node of Ranvier to convert direct current to the looking like an alternating current within the body. So that means even we are working on alternating current only. The current that will make us shock, the same current is there in our body. Our neurons are exchanging the information in the form of AC only, but we are not directly saying AC. Okay, but that is how we are measuring our body's electricity in the form of volts. Okay, volt is for AC current. Okay, I don't know much about physics, but this is the ideology of having this
threshold, stimulus, etc.
Yeah, thresholds. Yeah. So yeah, this is all about the nervous cell and thing. And coming last and finally is the neuroglial cells. These are the cells that supports the actual nerve cells. We have ependymal cells responsible for CSF secretion. Oligodendritic cells, they are responsible for nurturing the neurons. Satellite cells will ensure to control them. They will ensure the microenvironment of the neurons, then astrocytes, they will do healing of the neurons. Okay, if any neuron damage, then it will try to heal the neuron, it will try to regenerate the neuron. Then microglial cells, they will give the micronutrients to the neural cells. And Schwann cell is responsible for the myelination of the neurons, the axon of the neuron. Okay, so this is all about the cells and their significance. Thank you very much, students. If you have any doubts, you can ask me. If not, we can end this session. Any doubts in today's lecture?
Yes, sir.
Yes, Mr. Raman.
Sir, as we have studied that the cardiac cell and neuron cell, they cannot be divided, they cannot be regenerated.
Yes, yeah.
Sir, then how the astrocyte will help in regenerating the neurons?
That is a very good question. There is a limit of regeneration. If there is a there is an axon and the myelin sheath is damaged, in such case, a microcyte will try to it will try to compensate. There is a limit of repairing. Beyond that limit, no, they cannot unrepairable. Actually, that is correct.
Okay, sir. Thank you, sir.
Yes, to an extent, our body have even
Yes,
the cardiac cells and neural cells have the tendency to regenerate. Okay, to an extent, not to completely. Yeah.
Okay, sir. Yes, sir.
Any more questions? I think no more
Hello,
questions.
sir.
Yes, Mr.
Yes,
Raman.
sir. Is our MST will be online or offline?
I'm not sure actually. There is no clarity on it. Most probably it might be. I'm thinking, okay, 60% online, 40% offline. So I don't want to give any definitive statement on it. I'm not sure.
Okay, sir. Thank you, sir.
I think to my knowledge, it will be better prepare for offline. Why online?
Yes, sir. Offline mode is better because in online mode, some students can do they do cheating.
Anything can happen. Even in offline, cheating can happen. We cannot say only in online, but yeah, the offline will give more satisfaction compared to online. Yeah, that's
Sir, from third, it will be offline.
Most probably, I'm not sure.
Almost.
Sorry, guys.
Almost prepare for offline. Okay, why you want online? I don't encourage it. Okay, don't encourage online exam. Okay, yeah.
Sir.